Okay, nag-record na. Okay, so the title of the game is Stick to Word. You just have to guess the specific word using the two pictures that are presented. Okay, so let us start. So the first um, two pictures ito. So ano ang word sa ito? Okay, both of us,
Hotdog.
either
Hotdog.
wow, speak boy, can something great in that. Actually, wait, may guess sa answer. Di kaya makita sa akong kuan. Ang disadvantage sa Google Meet. Philip, yeah, you're correct, Philip. It's hot dog. Okay, next. Sayang ka, Madeni. Kani, what's the word? Any guess?
Lobat.
Ha? Lobat? No.
Sleepy.
Ano ka? Sleeping. Sige, unsa
Low
man.
bat. Low bat.
But no, hindi. Any other guess? Sige, walang surrender na. So, ang correct answer is lay off. Nakalayman ang tao. Kanya, off
Yeah.
on button. So, off button. Lay off.
Uy.
Okay, so get ready now for the third picture. Kani, so na siya kuan. Hint.
Mouthwash.
Mouthwash.
Correct. That's so easy. How about this?
Light bulb.
Yes, correct. Light bulb. And the last picture, last na di siya class, kaya para dili ko tayo magdugay aning dapita. Kani, what's the word?
Playlist.
Unsa? Na graph.
Graph base, ma'am.
Graph base? No. Other term for graph. Other term.
Line base.
Database.
Yes, database. Very good. Yeah. Okay. So, yun naman din kung ano niyo. So, easy lang. So, next time na po kayo magpag-game na po ta aron ma-motivate po ta panagsanok. So, next time magpakuan ako, maghatag na ko og prize. So, kana para namoy load para madagdagan po ang inyong mga data sa inyong internet. Okay, so thank you so much for participating, class. So let us now go to the proper discussion. So our exercise number 2 is entitled the solid state fermentation. As you can see in the picture, these are earthenware flasks, and it has these are examples of solid state fermentation setup. So there are solid substrates inside, and it is covered with solid cotton plugs, and of course they have to cover it with papers and then seal it with a rubber band so that there will be no um, other contaminants that might um, grow inside the flask. Okay, and there are um, microorganisms that are inoculated in that flask. Okay, so let us define now what is solid state fermentation. Now, the solid state fermentation is the common technique and has been already used and practiced um, from the ancient civilization. So most of these products that are already produced by the solid state fermentation are the tempeh, the soy sauce, the annatto, the miso. These are Japanese products, ang miso, ang annatto, ang tempeh in Indonesia. So, and then fermented rice, and then the Japanese wine, which is the sake. Okay, so um, let's continue. So the process of SSF is performed on a solid substrate. So based upon the term or the name of the word, the solid state fermentation, so the substrate is solid. Okay? Well, admit na please ka. Okay? So mo po ang disadvantage kay mga concept. Okay? So with a low moisture content. So in compare with the submerged fermentation, so it is the microorganisms are grown in a liquid media. Okay, so it has water. It has many liquids that are mixed. Okay, so the substrates are dissolved in the submerged fermentation. But here in the solid substrate, so there is only a low moisture content. And the advantage of this, it is a high product concentration, but only a relative low energy that is being required. Okay, so because there is a minimal diffusion and mixing of the product and also the biomass. Okay, so because mostly the products are liquid, okay, are in liquid form, diba? Ang substrate kay in solid form. So mas pansiya ka ng it can only be decanted right away, di ba? So, kung iyan ang biomass niya. Okay, the required content or water content in SSF is absorbed by the substrate in a solid matrix. Okay, and it offers more advantages of the growth of microorganisms for the transfer of oxygen because it has lots of um, solid particles or solid compounds. That it is not high in concentration, so there is more oxygen that can be circulated inside the media rather than the um, submerged fermentation. That is, submerged fermentation are regularly the aeration or the dispersion are really monitored with and still with siya every now and then. It's really constantly stirred. And you can also, there are also, when we conduct submerged fermentation in a early meal class, we have to incubate it in a shaker incubator so that it will be, there'll be shaking movement in the fermentation medium for 24 hours or for a particular time that your product will really produce. Okay, because of its efficiency and lower production costs, it is commonly used for the wine fermentation of root crops as substrates. Okay, so the advantages of this solid state fermentation, it is simple because we just have to for later, we'll just discuss with you the example that we do when I was a research assistant in the Philippine Root Crops Research Training Center. So we did solid state fermentation for the Taro's wine. If you are familiar with that, that's one of the products of the field crops, and it is still um, being constantly produced. Okay, so um, it is also cost effective because it is not that really the substrates are not that really pricey, and you can use also substrates that are really byproducts from the sugarcane production, yung mga molasses, yung mga rice bran, mga wheat bran, ganun. So these are you can really utilize it so that there will be zero waste, no? So third naman is less effluent release, and it also reduces pollution. So it also have high titers of the concentration of the product. Okay? But it also depends naman, may factor din na siya sa microorganism, depending on the microorganisms na siya. If it really favorably um, grow in low moisture content substrate. Okay, and also the aeration process is easy because it is low moisture content and the oxygen can really be accessed by the microorganisms easily. Okay, and second to the last is it resembles the natural habitat of some fungi and bacteria. If you know, if you remember in your microbiology class that many fungi and bacteria are really favorably grow in a low moisture content, di ba? So, and also yung mga um, food products, the number one, most of the microorganisms that grow first in the onset of foliage, um, like for the cookies, like for the mga mga jams, because they have a low moisture content, so they really fungi and bacteria will really easily grow there. Okay, so and the last advantage is that it has an easy downstream processing. So do you know what is a downstream processing, class? So it is the part of the fermentation that you have to separate the the medium and also purify your product for extraction and purification so that it will be readily distributed to the market if it is marketable, or if it has significant use sa society. Okay, that's the part you. So in this technique, just have to um, you just have to decant. Maybe there is a decantation process there because the substrate is already solid, and if your product is in a liquid form, you don't have hard time to centrifuge your product because there is just a ano man, lahi-lahi man ang form sa imong substrate uh, and product, and there is wala siya na mix kaayo gyud. So what I suspect naman that there are also products that are still in the substrate, okay? The substrate in the solid form. So it's just that na marami din talaga ang na hiwalay din talaga ang product because of that liquid form. Okay? So these are the examples of the substrates used in solid state fermentation. Example of these are sugarcane bagasse, tea waste. And most of them are really byproducts from sugarcane processing, mga sa tea production, the wheat bran, the sawdust, of course, that's it, apple pomace, and coconut oil cake. So sugarcane bagasse can be used also for the production of ethanol, for the production of PHB. It is a plastic that can be it's a biodegradable plastic that can uh, be used also to uh, to carry, or can be placed also to have for packaging of some food products or mga products, so that it's uh, environment friendly. So uh, this is one of the projects in the UPLB where I conducted my thesis. So in the environmental laboratory, so they use um, sugarcane baga. Okay, we have also apple pomace and then coconut oil cake. So okay, this is the product, one of the products of the Philippine Root Crops Research Training Center, the Taruz wine. So have you ever tasted this one na wine class?
Wala pa, ma'am.
Wala pa.
Root crop
So
niya,
let me
ma'am.
oh yes, this is a
Wala
gabi,
pa, ma'am.
VG9 na siya class. Mm. This is kanikod siya nga gabi wine class, the VG9 um, variety are also developed also in the Philippine root crops by our researchers there. Yon, I think Dr. Ferrarin is in charge in this in the breeding of the gabi. Yeah, so this is Dr. Ferrarin Ani. So ano siya, and then in the production of the Taruz wine, so the substrates are the sticky black rice. This is a sticky black rice and also the VG9. So what we did here is that we use this fermentation vessel. Okay, kani class mo na siyang among gikanda. One of the projects that I handled and experimented. So we use this fermentation vessel na kanina mo na siyang example din siya sa solid state fermentation. So nari siya quandary class. Actually, this is a glass na siya. Glass siya, kani siya kay metal ang iyakon. And then, ang ubos ani class, below here is a measured strain. Na siya strain in order so that when the fermentation takes place, so the liquid form, liquid product will just uh, will just pour diri uh, sa ubos, okay? So man siya nga diri man siya ang nakalocate ang substrates, and these substrates are the cooked sticky rice and then the VG9. So the VG9 are shredded, so they are peeled and shredded and also cooked. Okay, so along with the um, tar, this sticky black rice. So that's what the taro's one is named because of that taro, which is something gabi na siya sa kanang atong term, but it is called taro, yun na siya. So taro, and then this rice is, there is a, I don't know what language is that, it's called sometimes arroz, di ba, sa kanang mga arroz caldo, so it's a rice, so arroz means rice, so that's why it is coined from that, from the two words, so it is now called taro wine. Okay, so man siya class ang mitabo, so kana siya, so these sticky black rice are cooked, and then the VG9 is shredded, pour it out, mix it in the kanang it is they are cooked for mga 12 to 15 minutes, and then it is uh, cooled for a few minutes, so that after that it will now be inoculated with a microbial starter. So my microorganisms that are really used, these are called microbial starter. And this microbial starter is not just one kind of microorganism, but different types of microorganisms, such as yeast, such as bacteria, such as mold. Yeah, so mana siya ang involved. It's a um, multi-role, or uh, something like teamwork of the different microorganisms in order for the production of this uh, sweet wine. So, okay, so do you know that class, this VG9 is really kanang acrid and very kanang somewhat sa kanang katulgan siya nga kung imo la siyang lutuon ra and then you just have to eat it right away. So, but when it is processed, there is now no, wala na siya katul inom nimo sa Taros wine. So, yeah, that's the amazing part. That's the twist here that if it is fermented and consumed by the microorganisms or um, fermented by the microorganisms, the acridity of the VG9 will then be lost or then be kanang disappear, something like that. So, mana siya class ang nahitabo sa Taros wine. So, and then, yeah, so after mga, it is fermented or incubated for 7 days, and then uh, after seven days, the liquid form are harvested in class. So these are then pasteurized. The wine are pasteurized after, and then it is then bottled here in this particular. Then labeled it, put label in it, and then eventually it is the high mostly we sell it during anniversaries, gano'n. So, there are many available bottles during the anniversary with when it was still, face-to-face, meron pang, face-to-face na mga classes, gano'n. So, but then, now, so, I think I don't know the availability of this Atari wine now, right? No, depending siguro kung order lang. It's on the pre-order basis siguro. So yeah, that's one of the products. They are under the um, project of Dr. Julie Dutan. So that's my mentor. So she is an expert in fermentation technology, and she just retired this year. So um, that's it. That's the perfect example I can really relate with you. I can share with you about the solid state fermentation. So the microorganisms that are involved in solid state fermentation, so these are bacteria, yeast, fungi. It could be bacillus uh, species, Pseudomonas species. So they are aerobic microorganisms that can thrive in a solid state um, substrate, and also yeast, Saccharomyces cerevisiae, the Siliquaniomyces castile, and then also we have for the fungi Aspergillus niger, Aspergillus species. Okay, we have also Fusarium species, Mucor species. You know that Mucor is also one of the fungi that are involved in the fermentation of wine. Yeah. And we have also Penicillium notatum. So, the Penicillium notatum is the microorganisms that produce penicillin. Yeah. So that's the microorganism that was discovered by Alexander Fleming. Okay. Let's now proceed to the production of enzymes, the applications of this solid state fermentation. So, um, the application of these, it is used for the production of enzymes like the protease, the lipase, the cellulase, and pectinase. Okay, and we also have the production of organic acids such as citric acids and lactic acid. Citric acids are produced the citric acids are produced by Aspergillus niger. Okay, that is one of the microorganisms that are commonly used for that production. And third, it is also used for the secondary metabolite production. Could be antibiotics, could be quinolines, growth factors, and terpenoids. Okay, and we also have biocontrol agents. These are microbial agents, fungal agents, and for example, in the microorganisms, Liaganidium giganteum is a fungi that is used for the control of the mosquitoes. And the last one is it is used for the production of biofuel, and it is used specifically for the ethanol production. That is why class in Batangas, when we visited there for our tour, so sayang kay class na kanabang because of this pandemic, so naabay unta mo tour ani sa industrial chemistry. Yeah. So, matuunta mo kuan different um, mga processing plants, mga companies can go there in Cebu, and also some of the students went in Manila in UPLB. They had a tour there sa Erie, sa Biotech. But then now um, for our setup, so it would be hard that we should for the traveling part. So excuse me, class. I will just um, plug in my charger because it's low bat. So what is your where am I? Yeah, in the during our company visit or tour in our one of the subjects, so we went in a central azucarera where there is a production of the sugars in which they, of course, they have a sugar plantation and they use this sugar cane for the production of the sugars. And then the byproduct or the waste of these um, sugar cane are being collected by San Miguel. Yeah, because in a nearby town, there is it is located. The San Miguel Company Corporation is located. So then they have a collaboration that they have to collect the waste product or the byproduct of the uh, in their company, and then they used it as substrates for the production of their ethanol, okay, used for the disinfection that are already sold in the market. So we already it is really important nowadays because of this pandemic. So ayun, that's one of the examples of the production of biofuel. Yun. So let's now go for the objective of this exercise. Is number one, the only objective is to observe the natural fermentation of cooked rice through solid state fermentation. So this is just a natural fermentation without inoculating microbial starters, just like that. So it's just literally the spoilage of the rice. Okay. So the materials are just available at home, I guess, yeah, because you just have to cook any rice along with that sa inyong kanang ginakaon every day. So, and then you have to prepare 500 ml transparent glass jars. Just secure it, okay? And then you also need sterile used papers. How to do that is just you have to disinfect with alcohol, okay? So dili man nato siya pwedeng kanang masubmerge sa tubig. So you just have to disinfect it with alcohol. And then you also need spatula or just spoon. So yun. And then these are the preparation of the cooked rice. So along with your preparation of your rice for the meal, separate 2 cups of newly cooked rice and then let it cool. Okay, let it cool, and then after that, place the cooked rice into the sterile jar and cover it with a sterile paper. Okay, and then store it in a cool, dry place. Okay, that your siblings will not uh, um, damage it or will not no kanang dili siya hilabtan. You just have to secure it in a safe place, but in a cool, dry place. Okay, and then after that, you have to observe the glass jars every 24 hours for 7 days. Okay, how to observe? Just have to take a picture of your experiment every 24 hours, and you have to place it in your discussion. Okay, then observe the color, the texture, and the microorganisms that grow in the glass jars. Okay, the color of the microorganisms that are starting to grow in that particular day. And then, how about the other day? Ano bang nangyari sa rice? Yan. So, take down notes for your observation. So, these observations are to be discussed in your lab report. So, before doing this, placing the cooked rice into the jar, you have to sterilize your glass jars and your spoon. How to do it? You just have to cover the glass jars and spoon with aluminum foil. Then boil the glass jars and spoon for actually 30 minutes yun ang recommended. But because if you're using just an ordinary glass, dili siya mga Pyrex, dili siya mga kanang mga corny nga glass jar. So we just have to boil it for 10 minutes. So there's still sterilization that will happen. Okay. After 10 minutes, allow the glass jars to cool. And please do not use it when it's still hot. Okay. So observe regularly in your class na dili mo mapasok. You will not get burned. Okay. So this is an example of the picture of the result of the previous class that they did at home. So, mana siya ilang nakita ang observation. It's just the spoilage. So, at what particular day that they you have observed that there is already a growth of microorganisms and what happened to the rice? Are there any changes of the form? So, is it already watery? So, na ba siya liquid form? Ganun. So may mga colors sa microorganisms that already present at the top of the rice. So, So, inyo na siya i-discuss. Okay? So, in the conduct of the experiment class, okay, kani siya. So, you have to conduct it by pairs. So, you are, how many of you sa class? 18? 18 bang? 18 mo kabuk. So, we have 9 pairs. Okay? We have 9 pairs and only one of the pair will just conduct the experiment. No? So, di man siya individual. So, only one student lang will conduct because the other pair will just bang will help siya remind to observe and also accumulate also or collect the pictures and arrange it ganon. So yeah, so that's the teamwork of theirs sa inyo pair. Okay? So, and then you have 2 weeks to conduct your experiment. So, next week, I won't meet you for this so that you will have time. So, mas maayo class na kanang within that 2 weeks, because 7 days niyo siya i-observe. Okay? So, after 3 weeks, you have to submit your lab report. And of course, you have to submit it in the VSU EE. Okay? So, man siya ang for the exercise number 2. Do you have any question, class? Or clarification?
None, ma'am.
Okay, so for the pairing class, I will ask Jan Celestine to facilitate for the pairing. You can ask your classmates or pagsabot na lang mo on kisa ang mga pairing, kisa ang pairs para maka-communicate na mo dayon so that not all of you here are present right now. And after you have formed your pairs, please submit it to me or post it in the group chat so that I will take note of the pairing. Okay? But ang report is still individual. Okay? It is still an individual report. Okay? So, Jan Celestine, okay ra ni Mugang?
Okay ra, ma'am.
Okay. So, please post the pairing there is a group chat. Okay, so for the announcement class, so you have many quizzes to answer by next week. So you have a quiz for exercise 1 and 2. This is just one quiz. So, but the topic is are the exercises 1 and 2, and then another quiz for the lecture for module 1, because we are already finished with the module 1. So next week I will now proceed with the module 2. Okay. So I guess that's the last slide, and then the announcement. So napa mo may clarification, or how about for the methods? Nasabtan na ba ninyo? Is it clear with the class?
Hello, ma'am.
Yes, Philip?
Kanang ang kuan ma'am, ang rice hugasan pa ma'am.
Ha? Yes,
Ang rice
yes.
hugasan pa.
The normal way of
Cooking,
preparing your rice.
preparing.
Cooking, yeah. Yung hugasan. Yeah, so,
Thank you, ma'am.
sure. Um, what else? Yes, Jasmine? Do you have question?
Di man mo siya question ma'am ka lang sa ato bang schedule ma'am. Pwede
Yes.
8 to 11 na lang man kay naman may conflict sa 1 to 4.
Okay na. Di ako contact there.
So, kami
Okay, so na contact di ay sa other subject ni Jasmine. So, is 1 to 4. So, mag 8 to 11 na lang ta. Pwede ra po class. Okay ra na ako. So, I thought na walay conflict ani sa afternoon schedule. But it's okay. Mubalik ka sa 8 to 11. It's okay for me. Okay, so I'll
Thank you,
just,
ma'am.
okay. Sige, I'll just, announce it na naman sa group chat. Mabalik ka sa 8 to 11 na. Okay? So, any other things? Any other concerns? Okay, so if there are no concerns anymore, so I will now dismiss you with our class, and thank you so much, class, for listening. But before I will dismiss you, can you please turn on your camera and let's have a photo op. Okay, let's take a picture. Mga on Philip. How about the others? How about si Jaya, si Janessa, si Jasmine, si Rashid, and Krista? Okay, so basig wala dili pod functional ang ilang camera. So let's have a picture. Okay, let's wait for Janessa. Black. Okay. See. Okay. 1, 2, 3. Smile. Okay, another 1, 2, 3. Smile. Okay. Thank you so much, class. Bye.
Thank you, ma'am. Bye,
Thank you,
ma'am.
ma'am.
You're welcome.
Thank you, ma'am. Thank you, ma'am.
Bye.
Thank you, ma'am.
God bless.
Boy.